Hello, students. A very good morning. Give me a second. Yeah. Now the audio is working and the video is working. Sorry about it. Chalo. So, in today's class, we are going to be talking about one of the important sense organs that we have. Okay. So, that is the hearing. So, I know that it is a little bit of a complex topic, but I will make sure that it is portrayed as simple as possible so it helps you remember things. Okay. So, let's go. So we know that ear is very, very important for listening. Okay, we enjoy a lot of things with respect to hearing, especially music, okay, which is a gift of nature. So apart from that, the scoldings and the complaints and whatever it is all the bad things about hearing. Okay, and hearing gives us the sense of our environment. It will enable us, like human beings, it's all right for communication and for listening to things that we see or hear. But if you see in the wild, the hearing becomes one of the key aspects of survival. Because if you see the hearing capacities of different animals, they have exceptional hearing abilities compared to that of our humans. Okay, so when you talk about ears, okay, it is not just the organ of hearing; it is also organ of balancing. Okay, so let's talk about the anatomy of ear first, and then we'll go for the physiology as usual. So our ear can be divided into three parts. What are those? External ear, middle ear, and inner ear. Okay. So to make it easy for us to grasp things, all the brown things that we have, it's not brownish, it's not brownish, but it's a little bit of orange brown. So all this entire this portion becomes our external ear. All right. So sometimes what do we think? External ear is only what we can see, but it's not right. External ear also extends as a canal into our skull. Anyways, so this is the external ear, and then we have the pink portion. So this pink portion, okay, including this canal, it is called as the middle ear. And then finally, we have the blue portion, which is a very important region, that is the inner ear. Got it? So coming to external ear. First, we'll talk about external ear. Now, external ear is very important. Now, you might have heard about during the course of evolution, during amphibians, like especially the frogs, they start showing the presence of external ear pinna. So, similarly, what is the use of external ear is the sound waves that are created, okay, the sound that is around us has to be channelized into our eardrums, okay, it has to go amplified and channelized properly or directed properly into the inner parts of our ear. So that is what is the major role of external ears. So external ear has two important parts, that is pinna, or we also call it as ear pinna or ear lobes, this one, okay, and then you have the auditory meatus or ear canal, we call it as. So you can see here, this is our external ear pinna, okay, and then this long canal-like structure. So this long canal-like structure is called as auditory meatus, or it is also called as ear canal. Okay. Now, what did I tell you? The opening, you can see that throughout this external ear, you have one opening, isn't it? So that opening of the ear canal, you can see that there are some small hair-like structures or bristle-like structures. So these bristle-like structures or hair-like structures are quite important because it avoids any of the dust material from entering, okay, into the inner parts of the ear. Now, if you see the ear canal, so ear canal, they have a very important glands. So now you must be very, very aware of it because you frequently clean this ear canal. Okay, you can see that there is a waxy or suberin kind of a substance that is secreted into the ear canal. So the secretion comes from this gland called as, okay, ceruminous glands. Remember this. So ceruminous glands are the type of glands in this chapter they might not ask, but in structural organization, when it comes to modification of sweat glands, Okay, so it is very important. So cereminous glands are the names of the glands that are present in your ear canal. So these cereminous glands, what they do is they secrete secrete wax-like substance called as cerumen. Okay, so the cerumen is secreted, and ceruminous glands are the modification of sweat glands. So, along with the wax that is secreted, there is a cerumen, and the hairs that are present in our ears, they are both responsible for avoiding any of this foreign material from entering into our inner ears, okay, including the insects. Now, coming to the ear canal. So, if you see, we know that it starts from the ear pinna and it directs through the hole or the opening, okay, ear opening, and then you have a canal which we call it as ear canal, and finally it has to terminate at one point. So that point at which the ear canal ends, so this point is called as the tympanum. So you might have heard about tympanum a lot. So this is also called as eardrums. Okay, we also call it as tympanic membrane. Understood? So this tympanic membrane is where your external ear ends. If you see the eardrums or the tympanum, what you can see is this particular structure. Which one? So this tympanic membrane, what we see, this particular membrane actually has two sides, isn't it? One is the outer side, other one is the inner side of the membrane. Outer side is directed towards this side, and the inner side is directed towards the middle ear. So if you see, there is a thick layer of skin on the outer side, and then there is a mucous membrane on the inner side. So outside the membrane is made up of what? It is made up of skin. Okay, it is made up of skin. Whereas the inside, it is made up of what? It is made up of mucous membrane. Okay, got it? So this is about tympanum. They'll ask you the outside of the tympanic membrane because it's mentioned in your textbook and it becomes quite important. So the outside of the tympanic membrane is made up of what? It is made up of thick skin. Okay, it is a thick layer and it is a thick. It is made up of skin. Whereas the inner layer of tympanic membrane is made up of what? It is made up of mucous membrane, and it is a transparent membrane. Anyways, so coming to the middle ear. Middle ear is important. I will mention you what points you have to remember here. First, let's understand what about the anatomy of our middle ear. See here, this chamber that we have, okay, this chamber that we have. See, this is external ear. This is our eardrum. You can see here, right? This is our eardrum. And this middle portion, along with this tube that we have, they constitute the middle ear. So now what it leads to? Now you can see that middle ear is made up of one is tympanic cavity and ear ossicles. You know that this is the tympanum, and the tympanum opens into a cavity or a space-like structure. So this space-like structure is nothing but tympanic cavity and ear ossicles. So while we are studying the skeletal system, while we studied about the skull, I mentioned you that our ears also have three bones. Okay, malleus, incus, and stapes. Okay, so these three bones constitute the ear ossicles. Okay, now what is a tympanic cavity? Tympanic cavity is an air-filled chamber. Okay, so it is an air-filled chamber. So the role of this chamber is to separate your external ear from your internal ear. Okay, they separate your external ear and internal ear. And why it is air filled? That is becomes a very important point. You can see that there is a canal. Okay, so you can see that there is a clear canal or a tube-like structure that is coming from pharynx. Okay, so from pharynx, a canal extends all the way into the inner ear. So this canal is called as eustachian tube. It is called as what? Eustachian tube. So it is present on either side of your ears. So the role of this eustachian tube is to balance the pressure or it to maintain the pressure on either side. Okay, because the sound wave center and the air cavity is there. So the balance of this, you can feel the role of this eustachian tube while you deep dive into the water. Excuse me, or while you climb the mountains, when you go higher and higher altitude, you can see that because of the low pressure, your ear starts blocking. Okay, and you get a little bit of headache. So that is because of the eustachian tube. And the role of this eustachian tube is to maintain the pressure, okay, on either side of the eardrum. Got it? So coming to the ear ossicles, so you have to remember these names. What are these ear ossicles? Ear ossicles are nothing but the three bones that are interconnected with each other and which are present in the middle ear. Okay, so what are these three small bones that we have? As I mentioned, it is malleus, second one is incus, and third one is stapes. Okay, and you should understand this is the malleus. Okay, this is malleus, this is the incus, this is the incus, and this is the stapes. Okay, now see where malleus is attached. Malleus is attached to the, see, malleus is attached to what? It is attached to the eardrum, eardrum, or the tympanum. Whereas the stapes, where is it attached? The stapes is attached to the oval window. Okay, this we call it as oval window. Okay, stapes is attached to the oval window, whereas the malleus is attached to the tympanum. Understood? So you have to remember this. The malleus, incus, and stapes, they make up the ear ossicles. Okay, so I told you malleus is attached to the tympanum, whereas the stapes is attached to the oval window of the inner ear. Okay, entry of the inner ear, you can tell. Understood? It is also called as fenestra ovalis is another name for this, fenestra ovalis, or the oval window we call it as. Understood? So there was a question, which is the smallest bone in the human body? So the answer for this question is stapes. Understand this? Okay. Stapes is very important for us to remember. And the reason is because it is one of the smallest bone in the human body. Okay, as a factual thing to memorize. Got it? Chalo. Now, the most important part is we have to understand what is inner ear. Okay. So now when it comes to inner ear, remember here, while I look at the inner ear, you just have to look at this picture. And in this picture, what you can see is you can see that there is two different colorations. Okay, there is a purple and there is a blue color. Understood? All the purple color that you can visualize here, that is nothing but bony labyrinth. Okay, what is it made up of? Bony labyrinth. And all the blue color that you can see, that is made up of membranous labyrinth. Very important thing. Okay, so what is inner ear? Inner ear is made up of two important parts. Okay, or it consists of two important things. What are those? One is a bony labyrinth, other one is the membranous labyrinth. It is very hard for us to imagine it. So this is the best picture that you can use it to visualize how the bony labyrinth and the membranous labyrinth are organized within our inner ear. Got it? See, there is a blue and purple colorations. Just look at it. Now, what is this bony labyrinth? Okay. Now, bony labyrinth, we will just take a cross section, okay, and then look at it. Anyways, so when you look at the bony labyrinth, they have a cavity or a space, okay, which is filled by a fluid called as perilymph. Got it? Whereas the membranous labyrinth, they consist of two very, very important parts. What are those? One is cochlea, other one is called as vestibular aperture. You would have heard about cochlea. Okay, cochlea is the organ of hearing which help you to listen to things, whereas vestibular apparatus is one thing that helps you to maintain balance or equilibrium, I can tell. So let's look at bony labyrinth and membranous labyrinth, okay, and understand them. Got it? So we understood that we have bony labyrinth, all this bony region, that is, you have this purplish color. So they have this cavity, and the cavity is filled by what? Perilymph. Remember this. Whereas you have your membranous labyrinth. Membranous labyrinth can be divided, can that have two important parts. What are those? One is the cochlea, which is the organ of hearing, and then you have the vestibular apparatus, which is the organ of equilibrium. Done? Now let's go for the next one. Sorry. Chalo. So let's go for this inner ear, which is the cochlea. So cochlea is a coiled structure. You can see here, right? So it is quite coiled-like structure. Now what we will do is we'll take a cross section of the cochlea. When we take a cross section of cochlea, what you can understand is 1, 2, 3. Okay, how many are there? There are 3 canals. okay, which is present in the cochlea, which is quite coiled structure. Understood. So now we have the upper structure, and the upper structure is called as scala vestibuli. It's written here. So the upper structure is called as scala vestibuli. And then you have your middle ear, right? You have the middle portion, and the middle portion is called as scala media. Okay. So I'll just mention it here. It is called as scala media. So this one is called as scala media. Understood? Next one. Sorry. Yeah, scala media. And then you have your third lowermost portion that is called as scala tympani. So this is called as scala tympani. So see the spelling here. So what you can see when you take a cross section of cochlea, you'll come across three canal-like structures which are supercoiled. So those canal-like structures. Okay, what are those? The first one is called as scala vestibuli, which is in the upper part. Okay, and then you have the middle canal called as scala media, and the third one is called as scala tympani. So these three things. Okay. Now look at here. If you look at, sorry, if you look at your scala vestibuli, okay, you can see here, this is the scala vestibuli, right? So this is the scala vestibuli, and then you have the scala media. Okay, you have the scala media here. Now what you can see is there is a membrane that is separating your scala vestibuli and scala media. So the membrane that separates your scala vestibuli and the scala media is called as Reissner's membrane. Remember this? So the membrane that separates the scala vestibuli and scala media are called as what? Reissner's membrane. Now again, if I look at my scala media, okay, my scala media, and then my scala tympani, again, there is a membrane that is separating. You look at this membrane, very important membrane. So this membrane, okay, which separates my scala media and scala tympani is called as basilar membrane. Very important membrane. Both are important. Reissner's membrane is also important, and basilar membrane is also important. Just remember this. So how do you remember? You have the upper layer and the middle layer. So upper layer is called as scala vestibuli, middle layer is called as scala media, and they are separated by Reissner's membrane. Whereas you have the middle layer, that is scala media, and the lower layer, that is called as scala tympani, and they are separated by what membrane? Basilar membrane. Got it? Chalo. Now, if you look at your scala vestibuli and scala tympani, okay, these two regions, okay, do they have a cavity? Yes, they have a cavity. If they have a cavity, are they empty? No, they're all filled by this fluid, which is called as perilymph. Got it? So we heard about this perilymph called as bony labyrinth. Now you understood where the bony labyrinth is making it up. Okay. So scala vestibuli and scala tympani are made up of what? It is filled with perilymph. Whereas the scala media, so this portion is filled with what? Endolymph. Okay. This is filled with what? Endolymph. Clear? Now, coming to our the rest of the details about our cochlea, what you can see is we have the basilar membrane. Okay, this is the basilar membrane you can see here, correct? Now on the basilar membrane, you have some of the cells. Okay, you have the some of the cells. Okay, which are projecting. Okay, which are projecting. So these projections, okay, are into the scala media. So you can see here, this is the tectoral membrane. Okay, this is the tectoral membrane. Okay, and these are the stereocilia. Okay, these are the stereocilia. Okay, now what you can see here is to the basal membrane. Okay, basal membrane, and from the see, this is the resting on the basal membrane and projecting into the scala media. Okay, basal, you know what is basal membrane, correct? Correct, right? This is the basal membrane, this is the scala media, and this is the tympanum, correct, right? Now what it is doing, it is trying to project into the scala media. Okay, it is trying to project into the scala media. So these projections are nothing but receptors. Okay, these are called as receptor organs, and this receptor organ, so this receptor organ is called as organ of Corti. This receptor organ is called as what? Organ of Corti. So it rests on the basal membrane and it projects into our scala media. Got it? Now what does our organ of Corti consist of? Now, if you see the organ of Corti, what you can see is there are rows of cells that are arranged on the basilar membrane. Okay. So these rows of cells, what they have sensory hair-like structures called as stereocilia. What do they have? These rows of cells, okay, so are called as hair cells. So these hair cells, you can see here, outer hair cells, and then you have the inner hair cell. Got it? So what are they doing? They're trying to project hair-like projections, okay, above. So as they project upward, these hairs are called as what? Stereocilia. Understood? Now, as they move upward, what they do is, see, you can see here, these hair-like structures, they come in contact with this membrane, correct, right? They come in contact with this membrane. So this membrane is called as what? Tectorial membrane, okay? They come in contact with which one? Tectorial membrane. So they are quite important. The arrangement is quite important. We will understand why they are important, why it is significant. So understand this point. We understood that what is the organization of our cochlea. We understood that this is scala vestibuli, this is scala media, and this is scala tympani. Correct, no students? Okay. Now, what did we understood? This is the basilar membrane, and resting above the basilar membrane is the organ of Corti. And when I look at the organ of Corti, there are rows of cells that are arranged, and these cells, okay, are called as hair cells, and they project hair-like structures called as stereocilia. And the stereocilia come in direct contact with your tectorial membrane. If you see here, they're quite arranged in the order of their height, like this, somewhat like this, okay, from small to big. That is how they are arranged, the stereocilia. Understood? So you understood the organization of organs of Corti. Now we can go for what is vestibular apparatus. Till now we finished cochlea. The structure of cochlea is very easy. You understood? Now let's go and understand what is the vestibular apparatus. So remember, we understood. If I come back, we understand that inner ear is made up of bony labyrinth and membranous labyrinth. And when it comes to membranous labyrinth, we divide into cochlea and vestibular apparatus. When we come to cochlea, we understood the structure of the cochlea, and then this is made up, sorry, this is made up of perilymph, this is made up of endolymph, this is called a scala vestibuli, scala media, and we have the scala tympani. And then we understood there is a basilar membrane and Reissner's membrane. Above the Reissner's membrane is the organ of Corti, and the organ of Corti have rows of cells, and they have hair-like projections, and these hair-like projections are attached to this membrane called as tectorial membrane. Right? Now we have to understand what is vestibular apparatus, the second part of your okay, inner ear. So now when it comes to vestibular apparatus, what you can see in vestibular apparatus, this is the cochlea. We understood that. Now just extension of this region, you have one, you have two, and you have three, right? You have three semicircular canals. Okay, there are three semicircular canals, and what you can see is there is another portion called as Olith organs. Okay, so our vestibular apparatus contains two important parts. What are those? One is three semicircular canals, and then you have something called as Olith organ. We'll talk about it. Now, if you look at the two semicircular canals, okay, one is vertical and the other one is horizontal. Okay, so now these vertical canals, you can see this is all vertical canal, this is a horizontal, this is horizontal, this is vertical, isn't it? So if you look at these canals, vertical canals and horizontal canals, at the base of this vertical and horizontal canals, can you see that there is some lymph-like structure, like a bulge, okay, at the base of these canals? Each of them have a bulge-like structure, or we can call it as a lymph-like structure or a lump-like structure. So this is called as ampulla, okay, membranous ampulla. Okay, this bulging is called as what? Ampulla. Got it? And then sometimes, okay, it is also called as crista ampulla. Okay, that lump is also called as crista ampulla. So what is this? See, you have the lump, and inside this lump, what do we have is you have a long cilia-like cells. Okay, you know what is a cilia, right? Like hair-like structures. So cilia-like cells, and these cilia-like cells, okay, are present within the crista, okay, and they're grouped together in a bundle, okay, and this group together is called as copula. So what did we understand? We understood that if I look at if I look at my vestibular apparatus, it is made up of semicircular canals, okay, and then olith organ. So there are three semicircular canals, so which are arranged vertically and horizontally. At the base of them are the lump-like structures, which are called as ampulla. So inside this lump are crista ampullaris. What is crista ampullaris? So there are long cilia-like cells. Okay. So these cells are grouped together, and these cells which are grouped together are called are bundled together, which is called as copula. Understood? Inside it, what is there? There is a bundle of hair-like structures that are grouped, which is called as copula. That's it. Okay. Next one. If you look at what is olith organ, so if you look at olith organ, what you can see, olith organ is this one under this one. What is that? It is called as utricel and it is called as sacula. Okay, olith organ consists of what? It consists of utricel and sacula. Now, if I look at the utricel, okay, if I look at the utricel and the sacula, what do they have? They have a projecting ridge. Okay, there is a ridge-like structure like this. So this ridge-like structure is called as macula. Got it? So the ridge-like structure is called as what? Macula. Now you remember the hair-like structures that are grouped together called as a cristae. And then you have a ridge-like structure that is coming from the utricel and the sacula, which is called as macula. So these two things, which one, the cristae and the macula, they have a specific receptors. And the receptors of this, okay, which contain hair-like structures or the hair cells, okay, they are responsible for equilibrium, okay, for you to stand straight, stand left, lean right. How do you understand that you're doing this? Is because of the bending of these hair-like cells that are present in these cristae, okay, along with the macula. So that is what will help you to maintain the equilibrium and the posture of your body. Clear, students? Okay, I hope it is quite easy, and I hope you understood what I'm trying to teach. Okay, so chalo. Next one is the physiology of hearing. How do we hear? So it's quite easy. Now the sound waves enter through the ear pinna. Look at how beautifully the ear pinna is shaped so that it grabs the surrounding sound waves and then channelizes into our eardrums. So you can see that ear pinna varies in different organisms. You can see that all the prey, okay, all the prey like the deer or herbivore, they have a very long ear pinna because they want to hear even the slightest of the sounds that occurs in their environment. For example, you can see when the tiger or a cheetah or a leopard or a lion is hunting, they walk very stealthily and you cannot hear, like humans cannot hear at all. It's very stealth. Whereas these deer or antelopes, they pick up very faster because they have very huge ear pinnas, which gives them a broad range of sound waves coming into their eardrums. Anyways, so it enters to the eardrums and finally it reaches the tympanic membrane. To the tympanic membrane, it goes to the tympanic membrane. Now the tympanic membrane, what it does, it starts vibrating. Now once these vibrations of the tympanic membrane happens, now that will these vibrations are transmitted through these ear ossicles, that is malleus, incus, and stapes. Through the ear ossicles, finally it reaches the oval window. You know what is oval window, right? Opening of your inner ears. Now, once it reaches your oval window, now we know what is perilymph, right? Yeah, perilymph, which is present in the upper and the lower chambers of the cochlea. Okay, they also vibrate. When they vibrate, you can see this is the blue portion is the perilymph, right? So they start vibrating. So sorry, when the perilymph starts vibrating, now finally it reaches the scala tympani. Okay, so the bristles, the hair-like structures which are present on the scala tympani, they start to vibrate and bend because of the waves that are produced in the perilymph. Okay, so that will the ending of the hair cells press against your tectorial membrane. Okay, listen here. Okay, you had the basal membrane, right? So the basal membrane had hair-like cells, correct? And then there was a tectorial membrane, isn't it? So now, if you observe here, these vibrations, when they reach this point, these hair cells start moving towards a specific direction. So when they start moving sorry. Yeah. When they start pressing against a specific membrane, so the sensory hair cells get excited. So this excitation will reach the auditory nerves. Okay. So these impulses from the auditory nerve is taken to the auditory canal, auditory center of the brain. From the auditory center of the brain, you get the hearing. Yeah. Okay. So it all depends on the bending of the hairs that are present in the tectorial membrane. Okay, that is when the excitation starts. Okay, so next one we have is the olfaction, which is also called as organ of smell. Okay, basically, if you look at your nose, our nose, one of the best things that happens in our nasal canal, okay, or the nasal cavity, is that we have nasal mucosa, and the nasal mucosa produces a lot of mucus. Okay, this mucus is very very important because it helps the lot of chemicals that are present in the air. So the smell that comes to us is because of the chemoreceptors that are present. So the every thing that we have around that we can smell have the chemicals that are diffused in the air. So as we breathe in, so these small chemicals they dissolve with the mucus and they bind to the receptors. Okay, so which are called as olfactory receptors. Okay, so olfactory receptors are present on the cell called as olfactory epithelium. Okay, so they can understand the sense of smell, and they start getting excited depending upon the type of cells that we have. We have basically three kinds of cells that are present. So the neurons of this olfactory epithelium, you can see here, directly they extend to the external environment. Okay, and it is a broad bean like structures. Okay, which connects to the olfactory tract or olfactory bulb. Understood? So here these extensions are also connected to the limbic system. So when the chemicals get dissolved, you have your sense of perception of smell. And there are two things. One is the olfactory bulb, and then you have your limbic system. Both of them are involved in giving you the smell. You can see that when you smell something very fragrant, you have the pleasure feeling. When you smell the something very bad smell, you have the disgusting and vomiting sensation. So these are all emotions, isn't it? So that happens because of the limbic system. You should know that, right, already. So that is what it is. Now coming to the organ of taste, you have the gustation. So there are four tastes that you have. One is the sweet, salt, sour, and the bitter. So what you have, you can see here, your tongue is made up of taste buds. Okay. So there are taste buds. What are the different taste buds you have? You have circumvallate papillae, which is called lingual papillae. We call it as like lump-like structures. And then you have filiform papillae. We have fungiform papillae, and then we have we have one more, I think. Yeah, yeah. Anyway, we have circumvallate, filiform, and fungiform papillae. So what they do is, they basically are responsible for, the sensation of taste. Like how your olfactory epithelium, they dissolve the chemicals, and these chemicals are picked up by the receptors, olfactory receptors, and then sent to the brain. Similarly, you have gustatory receptors, okay, and then you have supporting cells. So these gustatory receptors are present on the taste papillae. Okay, these papillae, what I told, taste buds. So on each taste bud, there are hundreds of, you know, each papillae there are hundreds of taste buds. So they have very high surface area. So that will help you to grasp the taste while the chemicals dissolve along with the mucus. Okay, so now what will happen? Yeah, sorry. Now you have the sense of taste, and it goes to the gustatory receptors in the brain. Now the largest sense organ that you have for the contribution of sensation is your skin. Skin is the largest organ. So it contains several receptors for heat, cold, touch, pain, and then you have pressure, which is also called a somatosensation in total. Okay. So this is about your neural control and coordination. You have completed the chapter. So now we'll solve some questions. So if you have any doubts, students, you can just put it in the chat box. Okay. I'll just check it. Give me a second. If you have doubts, I'll just check the YouTube. All right. So now we will solve some of the questions. Let's see. Yeah. Unmyelinated nerve fiber is not enclosed by Schwann cells and hence not enclosed by myelin sheath. Enclosed by Schwann cells that does not form a myelin sheath. Not enclosed by Schwann cells but enclosed by myelin sheath formed by oligodendrocytes. Okay. Now, not found in the autonomous and the somatic nerve system. So you just read the question. Unmyelinated nerve fiber is. Okay. So when we talk about unmyelinated, remember that they are enclosed by Schwann cells, but they don't have any myelin sheath around them. Clear? So this is called as non-myelinated neurons. Next one. In human eye, the visual acuity is highest at macula, okay, lutea, optic discs, fovea centralis, optic chiasma. Okay, so these are your options. I've thought about it yesterday. So just look at it. In human eye, the visual acuity is highest at. So I told you, if you look at in the retina, okay, there is a spot, and this spot is called as the blind spot. And the blind spot is where you have the entry of your blood vessels and the exit of your optic neuron. And finally, lateral to the blind spot, there is a region called as fovea centralis, and the fovea centralis is yellow in color, and they are filled with cones. And here you have the highest visual perception. Here you can't see anything at the blind spot, but at the fovea centralis, you have the highest visual perception. So the option is option 3. Let's go for the next question. Select the incorrect statement regarding synapses. Impulse transmission across a chemical synapse is always faster than that across an electrical synapse. The membranes of the presynaptic and the postsynaptic neurons are in close proximity in an electrical synapses. Electrical current can flow directly from one neuron into the another neuron across the electrical synapse. Chemical synapse use neurotransmitters. So you should select the incorrect statement with respect to this question. Let's look at the question. So let's look at the first option. Impulse transmission across a chemical synapse is always faster than that across an electrical synapse. So is the impulse transmission across chemical synapses faster than the electrical synapse? No. Electrical synapse is very, very fast compared to that of chemical synapse. So the first option itself is wrong. Now coming to the second and third and fourth options, they are right, but let's look at them. The membranes of the presynaptic and the postsynaptic neurons are in close proximity in electrical synapse. This is right. I told you, whenever you have an electrical synapse, they are not completely attached together, but they're very close to each other, but the electrons jump. Huh? So electrical current can flow directly from one neuron to another neuron. Yes. So this means they don't need any neurotransmitters. Direct impulse transmission happens. Whereas in chemical synapse, we need the use of neurotransmitters. There is no direct impulse transmission. Correct, bro? Fine. We have the next question. Axons cannot have synaptic knob with synaptic vesicle, distal end is branched, and then synaptic vesicles have chemicals called as neurotransmitters and Niesel's granules. So let's look at the question. They're talking about axon. We know that axon terminates into synaptic knob, and synaptic knob can have synaptic vesicles, and the distal end, that is the end of the axon, can be branched. Okay, and then you know that the synaptic knob has synaptic vesicles, and synaptic vesicles have the chemicals called as neurotransmitters like acetylcholine, whereas the Niesel's granules are not present in axons. What are Niesel's granules? Niesel's granules are a type of endoplasmic, okay, endoplasmic reticulum. Okay, so what is the role of this? They synthesize protein. Okay, all the neural proteins are synthesized by your Niesel's granules. They're present in your cell body. Okay, which is quite unique to your neurons. All right, students, thank you very much. And we have come to close to this chapter. We come to close this chapter. Next, we'll go for chemical coordination, okay, and control. So I hope you have understood the chapter. If you have any doubts, please be free to text in the chat box of YouTube or in the Zoom meeting or ask your teachers. They'll get back to us and we can discuss about this topic. I have stressed on certain topics that you have to remember. Please go through it thoroughly, okay? And then you'll be all right. Okay. Chalo. Thank you very much, students. And we can end the coordination. Next, we can go for chemical coordinate.